Hi, I'm Phil Gallagher and I'm the director of the Applied Physiology Lab. I'm going to give you a tour of our facilities here. So right now we're in the main portion of the lab, one of many spaces that we have for the Applied Physiology Lab. Within this space we have our metabolic cart over here where we can test for VO2 max and see what your metabolic capacity is and your aerobic capacity is. And from this lab we have a bunch of other spaces that I will show you coming up. This is our procedures room. This is where we do muscle biopsies and blood draws. But if we're going to do a muscle biopsy or blood draw, the subject would probably be laying down on the table here. After we get the sample, we then will process the sample in this area right here. And then the other part of this lab space is set up for storage for all the things that we need, all the equipment that we need to do those blood draws and biopsies. This lab space is our smaller BioChem lab. We have actually two BioChem labs in the Applied Physiology Lab. We mainly use this space for the storage of chemicals. We do some prep work here. We'll make some buffers and reagents that we may need. On top of that, we do some cleaning of our equipment that we need for the metabolic cart. Also within this space, we have a refrigerator freezer. We have a centrifuge to spin down our blood samples. We have a hood. And we have an area to test for hematocrit and hemoglobin. This space is the larger BioChem lab that we have in the Applied Physiology Lab. We have an island in the middle, and we divided this island so that this side of the island is for proteins, any protein analysis. The opposite side of our island is for nucleic acids, and we're looking at DNA and RNA samples. We have various small biochemical tools that we use, microplate readers. We have a real-time RT-PCR system, Western immunoblotting, other SDS page analyses. We have a small histochem lab, which I'll show you here in a little bit as well. So this machine is our older workhorse real-time RT-PCR system. So we're looking at gene expression. We want to see, for example, pre and post exercise, if certain genes get upregulated or downregulated. We'll actually use this machine to analyze that. And then this space is our small histochem lab. For example, when we're doing fiber typing with mass and ATPase staining, we'll use the cryostat over here. It's basically like a meat slicer, so we'll get individual cross sections of the muscle fibers, and we can stain them to see which fiber type they are or they express. And we also have a stereo microscope along with another light microscope in the other lab. This stereo microscope allows us to tease out individual muscle fibers from that sample, and then we can do single fiber analyses of the fiber type. So my research is in two main areas. One is muscle physiology, so we'll look at signaling processes that happen, intracellular signaling processes that happen in muscle. For example, does exercise cause some signaling to happen that may increase metabolism or may increase hypertrophy or may even potentially cause inflammation? We also are looking at how a muscle itself might play a role with injuries, for example, ACL injuries, and one of my students is looking at how fiber type might play a role in that, as well as what sex you are, male or female. On top of that, we are looking at fiber typing, again, how that might change with different analyses. And then we're also looking at, with aging skeletal muscle, there's what happens from the signal that when your nerves tell your muscle to contract to the time they contract. There's a whole bunch of machinery that has to take place and a series of events need to take place. Well, we're looking at the machinery that may change as we get older. It's called excitation contraction coupling, and so we're looking at some of the proteins that are involved in that space between what's called the T-tubule and the sarcoplasmic reticulum. Do those change as we get older? Is that helping? Is the proteins that are involved in that process as we get older, are we losing those proteins which are normally there to help tether these machineries together? So those are the two main things we're looking at with muscle. And then the other area that I'm looking at is with the effects of exercise on the immune system. And there we're looking at T cells, how T cells become active as a result of exercise. And for the longest time, people thought that exercise, if you do high intensity exercise, it causes your immune system to be compromised. Well, we're not the first to find this, but we've been confirming that if you do really high intensity exercise, at least your T cells, they get amplified, they get active, they want to fight something. It's part of what's called the adaptive immunity, so they haven't been told what to attack, for example, bacteria or a virus. But they want to do something. They just become active, they proliferate like mad. The gene expressions that they express as a result of that high intensity exercise indicates that they are proliferating and want to do something, but they just haven't been told what to do. So we're going to follow that up to see what signaling is happening within these cells. Is that signal coming from the blood, or is it coming from within the cells themselves, as the fact that they're just going through the bloodstream? For example, shear stress might be a role in this. So we're looking at other factors that may be involved in activating these T cells as a result of high intensity exercise.
Hi, my name is Samantha Richter. I am currently in my final year of my master's in exercise science. I've been working in the Applied Physiology Laboratory since undergrad, so for about 4 years now. Currently for my project, what I'm doing is I'm looking into sex differences in fiber expression, more in the sense of implications for ACL injury. I personally have torn my ACL a bunch of times, so that's kind of what led me into this research. And now at the Applied Physiology Laboratory, I get the opportunity to look at fiber expression on the deeper level. So kind of what we do is we isolate a fiber or fibers from the muscle sample that we obtain from the muscle biopsy in the other lab. And then I take it here, I go through large format gel electrophoresis, and I just get a sense of what proteins are really involved and what proteins I'm kind of looking at. So myosin heavy chain is kind of the main ones.